Tyrannosaurus rex, the heaviest terrestrial predator that ever walked the planet Earth. One of the extremes of biological evolution, an animal so fascinating and yet so disturbing that it has captured and moved human imagination for over a century. But here's what most people don't realize. Everything you think you know about T-Rex is probably wrong, or at the very least, incomplete, because only recently have new technologies begun allowing us to truly understand how this animal actually lived. And what we've discovered shows that the reality of T-Rex goes far beyond just its size. Scientists just found something inside T-Rex fossils that should not exist after 66 million years. And I'm not talking about contamination or wishful thinking. I'm talking about actual blood proteins, real organic molecules that somehow survived since the Cretaceous period. But here's what makes this discovery even more mind-blowing. It's not just one thing, it's a cascade of revelations that are completely rewriting what we thought we knew about the most famous predator that ever lived. Because while scientists were finding impossible chemistry preserved in ancient bone, they were simultaneously discovering that T-Rex wasn't even alone at the top of the food chain. For over a century, paleontologists accepted a fundamental truth about fossils. Organic matter disappears completely over geological time. The process called diagenesis systematically breaks down proteins, DNA, and all other biological molecules, leaving behind only mineral impressions of what once lived. Bones become stone through replacement processes, and any trace of the original creature's chemistry vanishes into deep time. This understanding shaped how scientists approached dinosaur research. They knew they could study structure and form, skeletal anatomy, and biomechanics, but never the actual biochemistry of these ancient giants. The chemistry was gone. Forever. That was the rule. And then researchers began finding something that broke every rule in the book. Deep within the bone matrix of T-Rex and Brachylophosaurus canadensis specimens preserved in microscopic spaces between fossilized tissue were molecular signatures that resembled blood proteins, not mineral replacements, not contamination, actual organic molecules. The discovery defied every established principle about preservation limits, suggesting that fragments of 68-million-year-old hemoglobin had somehow survived the relentless march of geological time. The scientific community erupted in debate. Many experts dismissed the claims outright as contamination from modern sources or misidentified minerals. The idea was simply too radical to accept without extraordinary evidence. But then came the breakthrough that changed everything. The technique that finally proved these proteins were real is called resonance Raman spectroscopy. And if that sounds complicated, here's what you need to know. Scientists can now use laser light to identify molecular structures without destroying the sample. They direct specific wavelengths of laser energy at microscopic areas within fossil bone, causing molecules to vibrate at characteristic frequencies. Each type of molecule produces a unique spectral signature, creating what amounts to a molecular fingerprint that can distinguish between different chemical compounds with remarkable precision. It's like having a chemical lie detector that can tell you exactly what's there at the molecular level. The key lies in the 532 nanometer laser excitation wavelength, which specifically targets the iron-containing heme groups found in hemoglobin molecules. When this green laser light strikes authentic hemoglobin bonds, it produces a distinctive spectral pattern that differs dramatically from contamination or mineral interference. The technique can detect these molecular remnants even when they exist as tiny fragments embedded within the fossil matrix, providing conclusive evidence of their ancient origin. In other words, this isn't guesswork. This is hard, replicable science that proves beyond reasonable doubt that we're looking at real dinosaur blood proteins. But here's the question nobody saw coming. How is this even possible? How does organic matter survive for 66 million years when everything we know about chemistry says it should be long gone? The secret to this extraordinary preservation involves goethite, an iron oxide mineral that forms as hemoglobin degrades over time. And this is where the chemistry gets absolutely fascinating. As the original protein structure breaks down, iron atoms from the heme groups create protective mineral shells around protein fragments, essentially encasing them in a chemical tomb. This process occurs gradually, with the iron acting as both a catalyst for degradation and a preservative for what remains, creating stable molecular fossils within the surrounding rock matrix. Think about that for a second. The very thing that's breaking down the protein is simultaneously protecting what's left. It's like if rust not only destroyed metal, but also somehow preserved a snapshot of what the metal used to be. Iron-rich burial environments play a crucial role in this preservation process by creating chemical crosslinks between organic residues and surrounding minerals. These crosslinks stabilize protein fragments against complete decomposition, allowing certain molecular structures to persist far beyond their normal lifespan. The chemistry works because iron forms particularly strong bonds with organic molecules, creating hybrid organic mineral compounds that resist the usual processes of decay and dissolution. But before you start thinking we can extract dinosaur DNA and build Jurassic Park, let me stop you right there. This discovery carries important limitations that temper any hopes of genetic resurrection. While hemoglobin molecules can survive through iron-mediated preservation, DNA remains far more fragile and susceptible to complete breakdown over geological time. The double helix structure that carries genetic information cannot withstand the chemical assault of millions of years, even under the most favorable preservation conditions. Hemoglobin's simpler structure and iron content give it advantages that DNA simply lacks. So, no, we're not cloning dinosaurs anytime soon. But here's what we can do, and it's almost as exciting. The implications of preserved blood proteins extend far beyond just confirming that organic matter can survive. These molecular fossils represent chemical shadows of dinosaur physiology, providing direct evidence of how these creatures functioned at the cellular level. Scientists can now analyze oxygen carrying capacity, metabolic rates, and blood chemistry from actual dinosaur tissues rather than making inferences from bone structure alone. This chemical time capsule opens entirely new research pathways for understanding dinosaur biology that were previously considered impossible. By examining preserved proteins and their iron mineral complexes, researchers can investigate questions about dinosaur metabolism, activity levels, and physiological adaptations that we could never address before. The molecular fossil record provides a window into the internal workings of these ancient predators, revealing secrets that have been locked away for tens of millions of years. Want to know if T-Rex was warm-blooded or cold-blooded? The hemoglobin might tell us. Want to understand how efficiently it processed oxygen? The chemistry holds clues. Want to know how its blood adapted to the late Cretaceous climate? The molecules remember. It's like having access to a laboratory from 66 million years ago with actual samples from the most famous predator in history sitting on the shelf, waiting to be analyzed. But here's what makes this even more incredible. While scientists were unlocking these biochemical mysteries, they were simultaneously discovering that T-Rex itself wasn't what we thought it was, because the molecular discoveries represent just one piece of a much larger puzzle about late Cretaceous predator communities. The landscape of late Cretaceous North America was not the domain of a single tyrannical ruler. While T-Rex has claimed the spotlight for over a century, mounting fossil evidence reveals a far more intricate predator hierarchy than anyone previously imagined. The discovery centers around one of paleontology's most heated debates: the true identity of Nanotyrannus, a smaller tyrannosaur that scientists have argued over for decades. The controversy began with the fundamental question of whether Nanotyrannus represented a juvenile T-Rex or an entirely separate species. For years, traditional paleontology suggested these smaller specimens were simply young T-Rex individuals that hadn't reached full size. It made sense, right? You find a small tyrannosaur, you assume it's a baby version of the big one. Case closed. But several anatomical features didn't align with typical juvenile characteristics. The skull proportions were off, the tooth arrangement didn't match. The limb ratios created inconsistencies that made definitive classification nearly impossible using conventional analysis methods. Nanotyrannus possessed larger forelimbs, more teeth, fewer tail vertebrae, and distinct skull nerve patterns compared to T-Rex. These are features that are fixed early in development and incompatible with T-Rex growth patterns. In other words, if you're born with a certain nerve arrangement in your skull, it doesn't change as you grow. And Nanotyrannus had the wrong arrangement to be a baby T-Rex. But the proof? That came from an absolutely spectacular fossil discovery. The famous dueling dinosaurs fossil provided the key evidence needed to resolve this scientific mystery once and for all. This remarkable specimen, discovered in Montana, contained both a Nanotyrannus and a Triceratops locked in what appeared to be their final battle, preserved for eternity in stone. The exceptional preservation allowed researchers to conduct detailed analysis of the Tyrannosaur skull structure, revealing features that would prove decisive in settling the debate. Advanced cranial nerve pattern analysis exposed distinct neurological pathways that differed significantly from known T-Rex specimens. The optic nerve, channels, inner ear structures, and brain cavity proportions showed mature development patterns rather than the expected juvenile characteristics. Growth ring analysis of the bone tissue, similar to counting tree rings, confirmed that this Nanotyrannus had reached skeletal maturity at approximately 20 years old when it died, with growth rates indicating an adult specimen rather than a developing juvenile. The evidence definitively established Nanotyrannus as its own species, not a baby T-Rex. Not a sick T-Rex, not a stunted T-Rex, a completely separate species of Tyrannosaur that lived alongside the famous king. But here's where the story gets even crazier. Further research revealed not one, but two distinct Nanotyrannus species sharing the late Cretaceous ecosystem. Scientists identified Nanotyrannus lancensis and Nanotyrannus rex, each possessing unique cranial features, dental arrangements, and postcranial adaptations that distinguish them from each other and from T-Rex. The name Rex references the river Lethe from Greek mythology, the river of forgetfulness, acknowledging how this species remained forgotten for decades while being misidentified as juvenile T-Rex specimens. These discoveries meant that three different Tyrannosaur species coexisted during the Maastrichtian period, creating a predator guild system of unprecedented complexity during the final million years before the end Cretaceous extinction. Think about what that means. Three apex predators, all in the same family, all hunting the same general area at the same time. How does that even work? In modern ecosystems, you rarely see multiple large predators in the same niche. Lions and leopards in Africa partition their prey by size. Tigers and leopards in Asia avoid each other through different habitat preferences. So how did three tyrannosaurs coexist without driving each other extinct? The implications of this tyrannosaur diversity fundamentally altered our understanding of late Cretaceous ecosystems. Having three apex predator species sharing the same geographic range during the same time period requires sophisticated niche partitioning to avoid direct competition for limited resources. Each species likely specialized in different prey sizes, hunting strategies, and territorial requirements to coexist without driving each other to extinction through competitive pressure. Size differences between the three Tyrannosaur species suggest distinct ecological roles within the predator hierarchy. T-Rex, as the largest, likely focused on massive herbivores like Triceratops and Edmontosaurus, using sheer power to overwhelm heavily defended prey. The smaller Nanotyrannus species probably targeted more agile, medium-sized herbivores, relying on speed and maneuverability rather than brute force. This specialization allowed each species to exploit different segments of the available prey base without direct competition. It's like having three different restaurants on the same street. One serves steaks, one serves seafood, one serves pasta. They're all feeding people, but they're not competing for the exact same customers. The late Cretaceous Tyrannosaur ecosystem emerges as a sophisticated predator guild where specialized hunters fill distinct ecological niches. Rather than a simple monarchy ruled by a single apex predator, this period featured a complex web of predatory relationships that required precise behavioral and ecological adaptations. But here's the thing, the success of these apex predators depended on more than just size and strength. Their dominance required sophisticated biological capabilities that scientists are only now beginning to understand. The popular image of T-Rex as a lumbering, dim-witted brute couldn't be further from the truth. Behind those massive jaws and bone-crushing teeth lay one of nature's most sophisticated sensory systems, a biological masterpiece that transformed this apex predator into a living weapon of extraordinary precision. Brain endocast studies have revolutionized our understanding of T-Rex intelligence and sensory processing power. Scientists create detailed casts of the brain cavity by digitally mapping the interior skull features, allowing them to examine brain structure without damaging precious fossils. These endocasts reveal that T-Rex possessed sensory processing capabilities that rivaled modern apex predators, with brain structures optimized for hunting and survival in the Cretaceous world. The visual system of T-Rex operated like a precision targeting device. Forward-facing eyes provided stereoscopic vision that allowed precise depth perception when attacking prey, similar to modern birds of prey like hawks and eagles. The orbital positioning created overlapping fields of view that enabled accurate distance calculation, essential for timing devastating bite attacks. Unlike many other dinosaurs with eyes positioned on the sides of their heads, T-Rex evolved a narrow snout that wouldn't obstruct its forward vision, maximizing visual acuity during hunts. The optic lobes, which process visual information, were proportionally similar to those found in living birds, suggesting that T-Rex maintained exceptionally high visual acuity. This sophisticated visual processing allowed the predator to track moving prey across varied terrain, detect subtle movements that might indicate hidden animals, and coordinate complex hunting behaviors that required precise timing and spatial awareness. The visual system worked in perfect harmony with other sensory modalities to create comprehensive awareness of the surrounding environment. But here's what really sets T-Rex apart. Perhaps even more remarkable were the massive olfactory bulbs that gave T-Rex exceptional scent tracking abilities. These enlarged brain structures dedicated to processing smell enabled the detection of both living prey and carcasses across vast distances long before visual contact occurred. The size of these olfactory bulbs suggests sophisticated chemical detection capabilities that would have provided crucial advantages in locating food sources and identifying potential threats or competitors across the landscape. To put this in perspective, T-Rex had a better sense of smell than a modern bloodhound. It could probably detect a carcass from miles away, track wounded prey across changing terrain, and identify individual animals by their chemical signatures. This wasn't just a nose. This was a biological radar system tuned to detect anything edible within a massive radius. The integration of these sensory systems created a predatory package optimized for late Cretaceous conditions. Stereoscopic vision tracked targets with precision, while massive olfactory capabilities detected chemical signatures from considerable distances. Each system complemented the others, creating redundant sensory pathways that ensured hunting success even when individual senses face limitations from environmental conditions or prey behavior. T-Rex functioned as a living sensory laboratory, equipped with biological tools that represented millions of years of evolutionary refinement. Rather than relying solely on size and strength, these predators succeeded through sensory mastery and precise coordination of multiple detection systems. But here's where things get controversial. The sophisticated sensory arsenal reveals T-Rex as an intelligent hunter whose dominance stemmed from neurological advantages encoded in flesh and bone. Yet the true extent of T-Rex intelligence remains one of paleontology's most contentious debates. Early neuron count estimates initially suggested cognitive abilities that might support complex social structures, leading some researchers to propose that T-Rex was as smart as a primate. But these calculations have since been challenged as significantly overinflated. Research teams led by scientists like Kai Kasper have provided corrected estimates that place T-Rex intelligence firmly within reptilian parameters rather than approaching mammalian levels. Recent studies align T-Rex brain capacity more closely with modern crocodilians than with primates or other mammals. The revised calculations account for fundamental differences between reptilian and mammalian brain architecture. Recognizing that neuron density and processing efficiency vary significantly between these vertebrate groups,